So to sum up, I can say that mutation is any random spontaneous change in the base sequence. Since now we know that DNA is nothing but a polynucleotide strand, and every nucleotide has a single base, right? Because every nucleotide is made up of a base, a sugar, and a phosphate group. So if I say nucleotide sequence, it is the same thing as base sequence. So from a practical point of view, if we, just compare this definition with the definition that you guys did in O levels or IGCSE. So in O levels, it was a change in the gene structure, right? Or a change in the chromosome structure or the chromosome number. As I told you last time, that we are not concerned with the chromosome structure or the chromosome number as far as your AS syllabus is concerned. As far as your AS syllabus is concerned, we are more worried about this aspect, which is the gene structure. So when we say the structure of the gene is changed, we mean what is changed? The nucleotide sequence or the base sequence is changed, right? And that is defined as a mutation, and it's a random spontaneous change. Acha, from the point of view of this topic, the examiner or the syllabus wants you to put more stress on the topic of sickle cell anemia, keeping in mind that already when we were doing biological molecules, we did the structure of hemoglobin in detail. If I recap more, this is what I told you last time, that if we say hemoglobin, it is a globular quaternary protein made up of heme and globin. Heme is the prosthetic group where we have these, heme groups which will, over which you have iron atoms and their job is to bind to oxygen, right? Besides that, we have these 4 polypeptide chains, 2 alpha and 2 beta chains. Acha. When we talk about mutation, जब हम mutation की बात करते हैं और फिर हम उसमें example देते हैं sickle cell anemia का, so we are only worried about the change in the beta globin change. Nothing is wrong with the alpha chain. Nothing is wrong with the heme groups, only the beta globin chain. Since structurally it is a polypeptide chain, which means it's a sequence of amino acids, तो इसका मतलब ये है, हमने जो transcription and translation में किया, we all know that the sequence in which the amino acids are arranged depends upon actually the DNA code that was transcribed into an RNA codon, and the RNA codon that was translated into a sequence of amino acids. So agar, let's say we have an amino acid sequence for the adult hemoglobin, ठीक है, which is the adult hemoglobin allele, जिसको we give the symbol as hemoglobin A allele, ठीक है. फिर एक दूसरी allele है, which is the hemoglobin S allele, which is a sickle cell allele. So let's say if in my body, I apparently look completely normal. So of course, this means that I have the hemoglobin A allele, ठीक है. तो अगर मैं hemoglobin A को study करती हूँ, which is allele का मतलब gene ही है, तो that gene has such a genetic code or DNA code, which if I transcribe and translate, it will give me this sequence of amino acids, ठीक है. और अगर किसी को sickle cell anemia है, and if we see the sequence of the amino acids, which means if we zoom into the primary structure, we see that, oh, the glutamic acid has been replaced by valine, ठीक है. अच्छा. Also bringing some concepts of biological molecules that we've done before is, when I was teaching you amino acids, I told you that there are various kinds of amino acids, polar, non-polar, hydrophilic, hydrophobic, है ना, बहुत सारे types के. Acha, now glutamic acid is a polar amino acid, right? And the polarity of the amino acid is actually dependent upon what kind of R group that particular amino acid has. And when I say polar amino acid, Amna, what do I mean by that? Polar का मतलब क्या?
Soluble.
Soluble in water?
Yes, yes.
And non-polar means insoluble in water, right? So glutamic acid is a polar amino acid, and it's replaced by valine, which is a non-polar amino acid. So since we all know कि Alia, now you tell me, ये जो आपको protein नजर आ रहा है, which is the structure of the hemoglobin, ये, of course, quaternary तो ये by definition इसलिए है कि इसके पास more than one polypeptide chain है. But besides that, if I ask you, is this a globular protein or a fibrous protein? What will be your answer?
Miss, it's a globular protein
Why did you say it's a
because it's
globular
folded
protein?
into a spherical shape.
It's folded into a spherical shape. Acha, when I say globular or when I say fibrous, am I talking about its functionality or its structure? Yes, Alia.
Miss, functionality.
Okay, fine, functionality. तो फिर आप उसको make a connection. If I say globular, एक end पे मैं कह रही हूँ globular, and on the other hand, I'm saying that this globular nature of the protein is related to its function. तो कौन सा function of the hemoglobin?
Miss, globular proteins are soluble, but fibrous proteins are insoluble.
Very good. Globular proteins are soluble and fibrous proteins are insoluble. ये बात तो समझ आ गई. थोड़ा function के करीब आओ. Globular proteins are, they have a functional role or a structural role?
Miss, they have a functional role.
Okay, so give me make a connection with hemoglobin. What is the function of hemoglobin? To
Hemoglobin dissolves oxygen to form oxyhemoglobin.
okay, fine. Don't use the word dissolve. Dissolve वाला word नहीं use करना, because dissolve gives the examiner a wrong idea. You will say to use two words. ठीक है, यही words अब आगे भी use करोगे. Say hemoglobin attaches to oxygen. Hemoglobin binds to oxygen. ठीक है, attach करता है, bind करता है reversibly, and that's a function of the hemoglobin. Now, since hemoglobin is a part of the red blood cells, and since red blood cells are floating in your blood plasma, so by that perspective, hemoglobin should be water soluble. Should it be water soluble? Yes or no, Alia?
Yes, miss.
Of course, it should be water soluble. अब जब वो water soluble है, तो इसका मतलब ये है कि let's say if we talk about these globin chains, they should have amino acids of such a variety that all the water soluble amino acids should face outside, and all the water insoluble amino acids should face the interior of the molecule. Outside इसलिए कि ये जो outside वाला face है ना इस molecule का, it's facing the aqueous environment of your body. And when we talk about hemoglobin, तो जो aqueous environment आपकी body का है वो blood plasma. ठीक है? Why am I going into so much discussion? The reason for this is that when we talk about mutations, और जब हम कहते हैं कि the hemoglobin A allele is equivalent to having a amino acid sequence of such a variety, और hemoglobin S जब mutation आ जाता है तो amino acid glutamic acid, which is a polar amino acid, is replaced by valine, which is a non-polar. तो इससे होगा ये कि the solubility, just by the replacement of this one amino acid, the solubility of hemoglobin will be changed. When the solubility of hemoglobin is changed, so the shape of the red blood cell is also changed. Instead of the red blood cell having a biconcave shape like this, the red blood cell becomes sickle shape. Insoluble हो जाएगा, fibrous सा हो जाएगा आपका hemoglobin, right? So which is why we can see that such a small change. What is that change? The replacement of one amino acid with another amino acid is going to change the entire structure of the hemoglobin. फिर हम ये देखते हैं कि how does this change happen at a genetic level? So we said that we all know from our prior knowledge that DNA makes RNA and RNA makes protein. So there must be some change happening at the level of the DNA code that led to a differently transcribed mRNA, and which led to a differently translated protein, which is hemoglobin. ठीक है, एक चीज आपको याद रखनी है कि when we are talking about sickle cell anemia and when we are talking about mutations, always remember that this mutation is affecting your beta globin chain only. Ahmad, are you understanding whatever we are discussing right now?
Yes, miss.
No problems?
No, not yet.
Okay. If you have problems, you can always stop me, right? And I might not be able to see your hand raised, so you can simply unmute yourself. Okay, the same thing which I discussed right now. So very quickly, go through this and then I'll move forward. Just read the bullet points. Ahmad, can I ask you a question?
Yes, miss.
Okay, so here it says that reduced solubility of the protein as in the beta globin chain, alters the shape of the red blood cell, reduced ability to carry oxygen. Why do you think that a sickle-shaped red blood cell has a reduced ability to carry oxygen?
Because the reason is that जो उसका biconcave shape होता है,
Mm-hmm.
it has more like dense things so that it can absorb more oxygen it can bind with more oxygen. Whereas the sickle cell anemia shape is like thin, so I guess it binds with less oxygen.
Okay. And what about this one, the third bullet? It causes blockages in small blood vessels. Why?
Because the thing is that the small hemoglobin biconcave shapes are basically they just fold. Well, no, they just don't fold. They just compress, like when they pass through the small block of the small vessels. But the thing is that the sickle cell anemia, they're like elongated, so they can just
Get stuck.
preoccupy them. They can get stuck. Yeah.
Exactly. Now, see, when we say, look at this, this is a biconcave disc, right? And this is a sickle shape. The beauty of biconcave disc is that it can remain like this. It can become dumbbell shape like this. It can, it has a flexible sort of shape. It can change its shape. It can change its shape and accommodate itself so that the red blood cells keep flowing in the tiniest blood vessels, which are the capillaries. But it's not the same for sickle-shaped red blood cells. They get stuck, and especially they get stuck in the tiny capillaries. And such people who are suffering from sickle cell anemia, they might suffer tissue damage or tissue injury. Let's say if it's the blood vessels that are going to the brain, such people can die of stroke or myocardial infarction or even renal failure if it blocks the renal circulation, right? And if this is the same thing. Again, I'm going to repeat one thing again and again, that don't forget that this mutation is affecting the beta globin chain, not the alpha globin chain. And it's a mutation in which, and you have to remember the names of these amino acids, glutamic acid is replaced by valine. You also have to remember that glutamic acid is a polar amino acid, whereas valine is a non-polar amino acid, right? Yes, this is all repetition from last time, but I had to. So if we look at the DNA code, the DNA code is actually CTT, which is replaced, since we all know that our DNA code is a triplet code, which means it's a triplet of bases that is coding for one amino acid, right? So what is the triplet of bases that codes for glutamic acid? Let's do. For example, this is your sequence for the HbA allele, and this is the sequence of amino acids for the HbS allele. So what is that difference actually? ये जो है replace हो गया इससे, right? So CTT was the DNA code that was changed to CAT, which means which base was removed? This T thymine is being replaced by adenine. Just by replacing one base, the entire amino acid is changed, which means glutamic acid is changed to valine. Now, this brings us to now a new a new subtopic within the topic of mutation, which is types of mutations. So mutations can be of different types. The first variety that we are doing right now is substitution mutation. ठीक है, क्यों हम इसको substitution कह रहे हैं? क्योंकि हम क्या substitute कर रहे हैं? We are substituting one base with another base. It's a nitrogenous base. So we are replacing T with A, and this will change the entire sequence of the amino acid. और क्योंकि हम एक base को दूसरे base से substitute कर रहे हैं, that's why hence this kind of mutation is known as a substitution mutation, right? So you can say that the mutation that is responsible for sickle cell anemia is an example of substitution mutation, right? Acha, now Amna and Alia, can you tell me, I actually exactly don't remember, have I done this with you? I'll just put it up on the screen. Give me a second. Have we done the salient features of the genetic code? Ji Amna.
Yes, I don't think so.
No, we've not done it. Okay. Okay, so I think we should do this first so that you can understand fully what is happening over there, right? We'll just put a break on over there. At least we have now understood what a mutation is. How will it affect the development of sickle cell anemia? What difference happens when a polar amino acid is replaced with a non-polar amino acid? And what will be its final effect as far as the function of the hemoglobin is concerned? Okay. Keeping all that in mind, we should also remember that whatever we did in the structure of DNA, in DNA replication, in transcription and translation, we have to apply this onto the features of the genetic code. इसमें बहुत सारी ऐसे बातें हैं, ऐसी बातें हैं which you already know, but there are some things which have to be told to you. Number one is that all of us, whether it's humans or animals or fungi, bacteria, yeast, कुछ भी, except for viruses, we all share the same DNA code. क्या मतलब है इस बात का? इसका मतलब ये है कि the DNA code is universal, which means it is made up of the same 4 bases: adenine, guanine, cytosine, thymine. DNA की बात हम कर रहे हैं, ठीक है? Whether it's bacterium, even अगर हम बैक्टीरिया में कहते हैं ना कि इसके पास 2 न्यूक्लियस नहीं है, it just has a loop of DNA. Even if it has a loop of DNA and the DNA is naked DNA, it doesn't have its own proteins, but it is universal in that the bases are the same. एक ये बात. Second, every living organism except viruses, a triplet of bases codes for a single amino acid. ठीक है? हर living organism में, let's say, to give you an example, it's written ATC is representing 1 amino acid. GCC is representing 1 amino acid. That's a fixed rule. ठीक है? Okay. So, our DNA code is a triplet code. Let's look at the variety of the amino acids available in nature. Nature में या हमारी body में जो types हैं amino acids के, there are 20. ठीक है? So all this calculation has already been done, but just for your interest sake और कभी कभार आपके MCQs में भी आ जाता है कि ये सवाल उठता है कि let's say हमारे पास जो possible variety of bases है ना वो 4 है, right? We've got A, T, G and C. We don't have more than 4 kinds of bases as far as DNA is concerned, right? फिर हम ये कहते हैं कि जो total number of amino acids हैं in our body, those are 20. ठीक है? Now, although the textbooks have already told you that every 3 bases will code for 1 amino acid, but let's suppose we did not know कि 3 base एक amino acid के लिए को represent करते हैं या 2 base या 3 base. So let's say कि if we had to think about it, so we could think like this. ठीक है? Let's say if we say that 1 base represent 1 amino acid, तो इसका मतलब ये हुआ कि अगर हमारे पास 4 ही टाइप के बेस हैं तो we would have ended up in 4 amino acids. You can see I have done some probabilities over here. ठीक है? If I say, let's assume, I'm just assuming कि I have 4 possible bases. मेरे पास सिर्फ 4 bases हैं, and how many varieties of amino acids do I have to make? 20. So technically, if I assume that 1 base is representing 1 amino acid, तो अगर मेरे पास 4 bases हैं, तो 4 raised to the power 1 means I'll be ending up in only 4 varieties of amino acids. ठीक है? So I am rejecting this hypothesis. Let's move to another hypothesis. If I say, अच्छा मैंने reject कर दिया, अब ये करके देखते हैं. 2 bases, any 2 bases जैसे AT या CG are coding for 1 amino acid. So from that perspective, if I do a mathematical probability, 4 raised to the power 2, I end up with 16 amino acids. लेकिन that's not true because nature में या हमारी body में there are 20 possible amino acids, 20 varieties हैं. तो ये भी hypothesis हमारा reject हो जाता है mathematically. ठीक है. Third, which I am taking is that, okay, let's assume that every 3 bases are coding or representing one amino acid. Usme we end up with 64. Although 64 is a very large number as compared to 20, लेकिन ये जो हमारा third वाला hypothesis है ना, ये correct है. Why is it correct? I'll show you how this is correct through a table. I hope I can find this table. Yes, there's the table. Look here. You can see lots of codons over here. अच्छा. Amna, आप मुझे बताओ. What gives you an idea that these are codons and they are not DNA codes? इसमें एक clue है in this table. What's that?
They have you.
They have you. ठीक है? Alia, are you understanding this? कि uracil है, तो it has to be a codon and it's not a code. ये बात समझ आ रही है?
Yes, miss.
ठीक है? You नजर आ रहा है. अच्छा. Now, if technically they are codons, तो जरूर इनका कोई ना कोई कोड भी होता होगा. है या नहीं है? A DNA code. Let's suppose, okay, if I ask you, let's make a DNA code for the codon U. चलो इसका बनाते हैं हम. Let's make it U, and U. What will be the complementary DNA bases for U? Yes, Alia. U के साथ कौन complementary base pair करेगा? अगर हम इसको कहते हैं कि ये RNA है, इसका हम एक DNA बना लेते हैं.
So miss, it would be A.
It will be A. ठीक? अच्छा. ये तो मैंने सिर्फ इसलिए किया ताकि आपको समझ आए कि what I am actually trying to tell you. In this grid, you can see how many codons. Count करके बता दो कितने codons है if we count them. हर box में कितने हैं? 4. Yes? In every box you have 4. Let's count them. 1, 2, 3, 4. And how many boxes do you have in total? Total number of boxes kitne? 1, 2, 3, 4. 4 into 4, 16 boxes. Agreed? Guys, can you hear me?
Yes, miss.
Right? अब आप बात गौर से सुनो. हो सकता है कि आपको नहीं भी समझ आ रहा हो, but let's go slowly and it will be understandable. In this table we can see 64 codons. 64 इसलिए कि हर box में, in every box we have 4 codons and there are 16, so that gives us 64 possible codons. If we have 64 possible codons, this means they must have arise from 64 DNA codes. ठीक है? अच्छा. और इसमें हमें ये भी नजर आ रहा है कि there are more than one codon that codes for the same amino acid. कैसे? Let's zoom into it. For example, UUU and UUC both are coding or both are representing phenylalanine. ठीक है? फिर UUA and UUG both are coding for leucine and so on. So this actually gives us an idea कि ये इसलिए ऐसा होता है कि, for example, let's say if I have a mutation, ठीक है? Let's say कि किसी को by birth कोई mutation है for this, इसमें U is replaced by C, ठीक है? Instead of UUU, it's UCU. अच्छा. We needed the codon UUU to make the amino acid phenylalanine. So since we already have an extra codon which can also be responsible for the synthesis of phenylalanine, तो अगर आपके पास ये वाली mutation है, उससे कोई फर्क नहीं पड़ेगा because you already have a backup codon. ठीक है? तो बात कहाँ से शुरू हुई थी? बात ये शुरू हुई थी कि the total number of the amino acids in our body are 20. To make 20 amino acids, we have to have a rule. Now we rejected the 2 rules. What were those? We said that if one base stands for 1 amino acid, so we end up with 4 amino acids. If 2 bases are coding for 1 amino acid, so we were ending up in 16 अमाइनो एसिड्स, वो भी 20 से कम है। लेकिन अगर हम कहते हैं 3 bases are coding for 1 amino acid, तो we end up with 64 codes, या फिर आप कह सकते हैं 64 codons। और ये बात सच है, that's true, and this is actually the reality in our body, and that is why majority of us, even if we have mutations, तो हमारी body में पता नहीं चलता, because we've got these excess codons, which even if there's a problem over here or there is a, अब आप यहाँ देखो serine is an amino acid। serine के लिए कितने सारे codons हैं, UCU, UCC, UCA, UCG। So this means that even if there is some problem in this or in this, your body will still make serine because this codon is unaffected। So this brings us to the idea that although the total number of the amino acids are 20, but the possible number of the codons or the DNA codes that you have are 64. Okay? And this actually protects our body. This protects our body from, a blockage in the amino acid synthesis, because if protein synthesis aapki band ho jati hai, so we can die, because every little thing inside our body, ranging from hormones to enzymes to neurotransmitters to everything, it's protein. तो वो काम चलता रहता है, right? अच्छा, going back to where we were actually doing the salient features of the genetic code. So the first salient feature of the genetic code was, जो मैंने पहली बात की, कि चाहे कुछ भी हो, if it's an animal or a fungus or a bacterium or whatever, viruses के अलावा, all of us have a universal code. और जब हम कहते हैं कि हमारा कोड यूनिवर्सल है, इसका मतलब ये कि हम सब, every living thing on this earth, on this universe is made up of these 4 bases. Our code is universal. ठीक है? Our code is triplet. मतलब 3 nucleotides, or even you can say 3 bases, एक ही बात है, क्योंकि हर न्यूक्लियोटाइड में एक ही बेस होता है. They code for 1 amino acid. Third, our code is degenerate. ये जो मैंने अभी आपको पूरा बताया है ना, as far as this table is concerned, के the total number of the amino acids is 20, but the number of the codons is 64. इस property को कहते हैं degenerate. In fact, some textbooks also use the word redundant, कि our DNA is redundant. यानी बहुत सारा extra DNA भी हमारे पास पड़ा हुआ है, right? So this property is degenerate. और आपसे examiner पूछ सकता है कि what do you mean by a DNA being degenerate? So you will say that this actually means that we have got more codons as compared to the number of the amino acids. Or in other words, let me put it into better words, you will say that there are more than 1 codons which are representing 1 amino acid. And this is a protective function for us. Our body will not stop making amino acids if there is a problem. Okay? Yes, all 3 of you, have you understood this, or you're totally blank? Please tell me frankly. Understood. What about you, Alia, beta?
Understood.
Ahmed?
Yes, it's clear.
Yeah, good. Okay. And another thing is that DNA is non-overlapping and punctuating. Let's do that as well. Punctuating. Punctuation means full stops and comma and starting point, right? Our DNA code has a starting point and has a stopping point. How? When we were doing transcription and translation, over here, yes, the start codon, the RNA codon, was always AUG. And we used to say, I don't know whether I discussed this with you, लेकिन मैं अभी दोबारा से बता देती हूँ कि to remember that the start codon is AUG, we say AUG stands for August, and we say schools open in August. Schools start in August. So we'll say that the protein synthesis begins with the codon AUG. So इसका मतलब क्या है? इसका मतलब ये है कि whenever your body is doing protein synthesis, कोई भी protein बन रहा है, उसमें जो आपका start codon होगा ना, it will be AUG. ठीक है? And as per that start codon, this means that the first amino acid synthesized will always be methionine. तो क्योंकि ये एक rule follow हो रहा है, so we would say, what will we say? We'll say DNA is punctuated. Punctuated है ना, क्योंकि punctuation का मतलब क्या होता है? We begin a sentence with a capital letter. That's punctuation. We end the sentence with a full stop, and in the middle we put some commas. ठीक है? तो the start codon is always AUG, and the amino acid corresponding to the start codon is always methionine. Always. ठीक है? And of course, अगर आपको AUG का नहीं भी learn करना UAC anticodon, तो आप खुद बना सकते हो UAC anticodon, right? And then your body will keep on synthesizing proteins till it reaches a codon जिसको हम कहते हैं stop codon. और वो कौन सा है stop codon? यहाँ नहीं लिखा हुआ. I'll tell you. The stop codon is UR, UGA, and UAG. These are the stop codon. UR, UGA, and UAG. Right? Let's go again to where we were. जिसमें salient features हैं genetic code के. So yes, let's look at that at this table. Look, UR, UAG, UGA. Wherever you will see these, that's a stop codon. Okay. Amna, you tell me, why is it necessary to have stop codon?
In case we need an extra pair, maybe.
I can't hear you, beta.
Because I'm not really sure.
You know the answer. Look, your body is making different kinds of proteins. So do you think that if your body has received a signal to make, let's say, amylase, that's a protein. Insulin, that's a protein. Serotonin, that's a protein, right? LH, FSH, so many different kinds of proteins. So shouldn't they have a stop codon? Shouldn't the cell get a signal that, you know, now the polypeptide chain is complete? Yes? Right? So every protein needs to be very, very precisely made. Every protein should have a starting point and an ending point, which is why we can see that there are accurate, fixed, precise stop and start codons, right? So this feature or this property of a genetic code is known as punctuating. So we say our DNA code is a triplet code, number one. Our DNA code is universal. All of us share the same basis. Number two, our DNA code is redundant or degenerate. Number three, our DNA code is punctuating, number four, and our DNA code is non-overlapping, number five. Now, non-overlapping means that we can read the DNA code in a certain sequence only. How? Look here. Let's say if this is a DNA code, I'm actually zooming into a DNA code. So if I want to read it, so DNA is a double polynucleotide strand, right? तो अब मैंने आपको ये सिर्फ एक पार्ट ऑफ द पॉलीन्यूक्लियोटाइड स्ट्रैंड दिखाया, ये वाला दिखाया, लेकिन असल में अगर ये असली पूरा डीएनए होता तो यहाँ पे भी बेसिस होते, isn't it? Right? And we all know that the DNA molecule is a double polynucleotide strand, और जिसमें से एक स्ट्रैंड जा रहा है into 5 prime 3 prime direction and the other is in the 3 prime to 5 prime direction. तो which strand are both the strands readable? दो सवाल आते हैं. Okay, and second question is that how do we read it? ठीक है? When you want to read a story book, या आप इस वक्त मेरी स्क्रीन पे iPad पे कुछ चीज पढ़ सक रहे हो, so we have to follow a certain rule for reading. For example, if I want to read this sentence, अगर मुझे ये sentence पढ़ना है, write out the base sequence of the RNA form from blah blah blah. I have to follow certain rules. In English, I have to read from left to right. In Urdu or in Arabic, I have to read from right to left. And also that एक तो तरीका है कि direction same होना, सही होना चाहिए, and plus the words should be also written in a proper sequence. Similarly, if you want to read the DNA strand, there are two rules. ठीक है? वो rules क्या है? Rule number one, you can only read the strand in the 5 prime to 3 prime direction, number one. And you can only read it in triplet of bases and you can't overlap the reading. Kaise? I'll show you. For example, if I have to read this, एंड लेट्स अज्यूम कर लेते हैं कि ये 5 प्राइम 3 प्राइम है, राइट? अब मुझे इसको रीड करना है. आई रीड लाइक दिस: ATG, TTC, GAG, TAC, CAT, GTA, ACG. ठीक है? This is the only method of reading, and this is how, जैसे मैं पढ़ रही हूँ ना, मैं for example I don't read like this के I have to read like this, write out the. अब I don't mix this E with O or this T with this T. अगर मैं ऐसे करना शुरू करूँ, या मैं उल्टा पढ़ना शुरू कर दूँ, for example if I read like this, sequence based, the outright, that's also not going to make sense. So reading has to be done in a certain direction, and reading has to be done in groups of words. जैसे हम अभी ऊपर एक group of words पढ़ रहे थे. DNA की language में they are not groups of words, they are groups of triplets of bases. I can't read like this. For example, मैंने पहले पढ़ा ATG, and then if I read like this, let me use another color. पहले मैं पढ़ूं ATG और फिर मैं पढ़ूं GTT और उसके बाद मैं पढ़ूं TTC and then CGA and so on. I can't read like this. So this feature of the genetic code is said to be non-overlapping. So our DNA code is non-overlapping, means that it can only be read in triplets of bases from the 5 prime to the 3 prime end of the strand. ठीक है? Any questions? Please ask. In fact, I'll show you something from your textbook, where you have this strand that is shown to you, which you can read in a particular direction. Okay, let me show you. Okay, this is the one. Acha, in the new book, they haven't shown that strand, but still you can see the features of the genetic code. So a 3-letter code, right? The code is universal. And this means that universal ka matlab, this means that each triplet codes for the same amino acid in all the living things. Whether it's me, for example, let's say if UAC is coding for methionine, so this is the same, it's the same in a bacteria also, right? The code has punctuation, so we have stop signals and we have start signals, right? As I told you, TAC is representing methionine. Okay, Ahmad, can you tell me, if I say TAC is the DNA code, can you make the mRNA for TAC?
Yes, I don't know about this because we have not studied this whole thing.
Okay. Yes, Amna. Make the mRNA for TAC.
Miss AUG.
AUG. ठीक है? अच्छा. So A, याद है मैंने कहा था schools open in August. So AUG is the start codon. AUG is the start codon. TAC is the start, not codon, but start DNA code. And if I ask you, now this question is for you, Alia, make the anticodon for AUG. Anticodon for AUG. क्योंकि इसको कहते हैं हम, okay, this is DNA, right? This is the DNA code. DNA के लिए loves codon नहीं use होता, code use होता है. This is the DNA code. This is the mRNA codon. अब इसमें से आप anticodon बनाओ, आलिया.
Miss U
U
UAG UAC. Miss.
UAC. ठीक है? So this will always be the start codon, the start anticodon and the start DNA code, and all these code for the same amino acid, which is methionine. Then we did this property that the DNA code is redundant. Redundant भी आप use कर सकते हैं. Degenerate, which means if you want to explain to the examiner, you will say it actually means that some amino acids are coded for by more than one triplet. ठीक है, और इससे फायदा होता है. This is advantages for our body. And you will see this advantage as far as the topic of mutations is concerned. Coming back to where we were. अब mutations ज़्यादा सही तरह समझ आएगा. Let's look at this types of mutations. Okay. Now, the first type of mutation that we have done already is the substitution mutation. Or substitution mutation, agar exam mein aata hai na, don't even give it a second thought. Just quickly, without even thinking a lot about it, talk about sickle cell anemia. बस सबसे आसान है. आप लिख दोगे कि the DNA code CTT is replaced by CAT. So hence, by that definition, it is a substitution mutation in which the amino acid glutamic acid is replaced by valine. जिसकी वजह से, क्योंकि glutamic acid is a polar amino acid and valine is a non-polar amino acid, so it is going to change the polarity of the molecule, of the hemoglobin molecule. It will become less polar. The red blood cells will lose their biconcave shape and become sickle shape. ठीक है? अच्छा, ये तो हमने कर लिया सब्स्टिट्यूशन. अब जल्दी से ये कर लेते हैं डिलीशन एंड इंसर्शन. ठीक है? डिलीशन का मतलब अ बेस इज डिलीटेड, इंसर्शन का मतलब अ बेस इज इंसर्टेड. सिंपल. ठीक है? अच्छा, डिलीशंस एंड इंसर्शंस आर आल्सो नोन एज फ्रेम शिफ्ट म्यूटेशन. अब ये क्या है फ्रेम शिफ्ट म्यूटेशन? Frame shift mutation means that these are mutations in which the reading frame of DNA is changed. बिल्कुल अभी जैसे मैं आपको कह रही थी ना, for example, if I write like this, okay, let's say if this is the DNA code, I'm making a DNA code, right? CCT, CCG. अच्छा, I'm now deleting a base. Let me delete a base, and that is this one. Look at this very carefully. I'm deleting this base, and now I am rewriting the DNA code. ACT. C T C. Is the reading frame changed? Yes, girls. First, we were reading it like this. हम जब उसको पढ़ रहे थे, it was ACT, CCT, and CCG. आगे के bases मैंने नहीं लिखे हैं, but let me just write down some more bases. Let's say GGA. ठीक है, कुछ होते आगे के bases. So, if I have deleted the base C, I have changed the reading frame. Can you see that I have changed the reading frame? पहले मैं इसको read कर रही थी ACT. ठीक है, ये ACT है. अच्छा, previously I was reading it as CCT, but now the reading frame has changed to CTC. Has the reading frame been changed? Yes or no?
Yes, miss.
Right? When the reading frame changes, then the effect of that on the polypeptide synthesis or the amino acid synthesis is huge. It's big. Usually substitution mutations का effect भी huge होता है, लेकिन deletion insertion का effect बहुत ज़्यादा होता है because it changes the reading frame. जब ये बिल्कुल ऐसे है कि जैसे कि अगर मैं किसी paragraph में सही जगह पे if I don't put commas and full stops, it changes the meaning of the entire sentence. If I don't put a capital letter at the required place, right, it changes the entire meaning of a sentence. या फिर अगर एक sentence में से मैं एक word निकाल दूं, it changes the entire meaning. Similarly, over here, in case of an insertion mutation or a deletion mutation, the reading frame of the DNA will be changed. तो आप DNA को जब read करने लगेंगे, कुछ और ही उसका मतलब निकल आएगा. And मतलब निकलने का मतलब, it means actually in the real sense is that a different kind of a protein will be made, right? So it's a reading frame mutation. Let's look at this reading frame mutation. Now you can see a normal code in front of you. ठीक है? और उसके नीचे आपको you have a base has been inserted, and that inserted base is shown in bold, this one. Take some time, take 1 to 2 minutes to see how by inserting this base, जिसको मैंने red से highlight किया हुआ है, has changed the reading frame. अच्छी तरह से देखो. Because these questions are very challenging. when this reading frame was changed, because of this, it exerted a domino effect, and you can see TAG, 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 TAG has been changed to GTA, 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 and GTA, and so on. So you can see that just by the insertion of one base, the entire reading frame of the DNA has been changed. Another example, deleting a base. So even if you delete a base, then also the reading frame is changed, and such kind of mutations in which there is an insertion Guys, can you hear me?
Yes, Miss.
Acha, I don't know why I'm getting a message that I've been signed out. So I suddenly stopped. Okay, so this is the frame shift mutation, such kind of a mutation in which the, what do you say? There is an insertion or a deletion of the base. It's known as a frame shift mutation, right? All the triplets from the mutation onwards are affected, and all amino acids coded for will probably be incorrect. So the polypeptide or protein made as a result will be non-functional. Let's do a question so that this concept is strengthened. It's a very interesting question on the reading frame mutations, right? And I have it on my presentation. Let's do it. Okay, it's a challenging question, and it's a challenge for all of you. This figure shows, figure 4.2, right? The first 7 amino acids of the beta chain of the hemoglobin. Remember, we are doing the beta chain hemoglobin, not the alpha chain. Okay? First 7 amino acids aapko nazar aa rahe hain. The first amino acid in the sequence is valine. The 21 base pairs in the sequence of DNA that code for these 7 amino acids. So, what are you shown in this picture? सबसे पहले आपको first 7 amino acids नजर आ रहे हैं. ये भी नजर आ रहा है कि the first amino acid in this sequence is valine. This is the information number 2. And then you can also see the base sequence जिसमें 21 base pairs नजर आ रहे हैं. अच्छा, how come 21 base pairs? Alia, Amna, can you pick up those 21 base pairs? कहाँ हैं 21 base pairs? I can see 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14 triplets of bases. If I can see 14 triplets of bases, does it mean that there are 21 base pairs?
Miss, there are 14 codons.
Achha,
And one codon has 3 bases, so each base will make a pair with each other.
correct. But what word did you use? It's good that you made this mistake. You said there are 14 codons. Is this statement correct? Alia, think about it. What did you say? स्क्रीन पे देखो, ये आपको कोडॉन्स दिए हुए हैं या आपको कोड्स दिए हुए हैं, ये बताओ. Are these codons or are these codes?
Miss, they are codes.
They are codes. Why are you saying these are codes?
Because Miss, just codon and anticodon both given.
नहीं, गौर से देखो और गौर से देखो. Are these codons or are these codes? अच्छा, code का मतलब क्या? Code means DNA. In other words, you know what I'm asking you? I'm indirectly asking you. I'm testing your knowledge ये देखने के लिए कि आपको फर्क पता चल रहा है DNA और RNA में. If I'm saying it's a code, इसका मतलब I'm asking you क्या ये DNA? If I'm asking you it's a codon, so in other words I'm asking you is this RNA? So you tell me, ये जो आपको नजर आ रहा है, are these DNA or RNA?
Miss, it's DNA.
Very good, they are DNA. So these are not codons. So now use the correct terminology. बहुत गौर से आप अपने क्वेश्चन में देखो कि आपको codons दिए हुए हैं या codes दिए हुए हैं. So you've been given 14 triplets of DNA codes and you've been given 21 base pairs. 21 base pairs का मतलब कैसे? This is a base pair, CG, 1, 2, sorry, ऐसे करके. Let me just zoom into it. This is a base pair, ठीक है? One base pair, second base pair, third base pair. तो अगर हम सबको काउंट करेंगे तो ये हो जाएंगे आपके 21 base pairs. ठीक? अच्छा. Let's go forward into the question. Table 4.1 shows the triplets of bases that code for 7 amino acids. ठीक? Using 4.2, using figure 4.2 and table 4.1, table नीचे आ रही है. State what will happen to the sequence of the amino acids in the first part of the beta chain of hemoglobin. Now I'm, holding my screen like this so that all of you can see. Can you see the entire page, all of you? And is it clearly visible to you? थोड़ा सा मैं zoom कर लेती हूँ. अब आप देखो. Use this table जो नीचे दी हुई है and also the figure both to find out what will happen if the base pair at position 6 is deleted. Base pair at position 6. Answers to be seen from here. Position 1, position 2, position 3, 4, 5, 6. I've given you a lot of hints. G, think about it. Amna, can you think about it? Base pair at position 6 is deleted. Okay, this is the base pair at position 6. GC is deleted. If GC is deleted, try to see what code of DNA you end up with. Is the reading frame changed or is it not?
Yes, it is.
It is. Acha, theek hai. How is it changed? Okay. Let me show you this. Look at the way I've solved it and also look at the table. Let's look at the first DNA code. The first DNA code is GTG. ठीक है? GTG को हम यहाँ देखते हैं. GTG is equivalent to the amino acid valine. ठीक है? So nothing has happened to our GTG wala code. So hence this amino acid will be made. एक तो ये चीज, ठीक है? What have we deleted? We have deleted the base pair at position 6. Position 6 का मतलब 1, 2, 3, 4 and 5. हमने ये निकाल दिया. ये पूरा हमने base pair हटा दिया है. जब हमने base pair हटा दिया और आगे का जब हमने bases देखे ना, हम हटा के हमें जोड़ना है इसको, right? So see, it was GTG. Even if we remove this and if we move forward, तो दोबारा से क्या हो जाएगा? GTG, कोई फर्क नहीं पड़ेगा. And even if we remove this C, तो आगे भी C है तो वही हो जाएगा CAC. So this means that if we look at CAC, what amino acid is it representing? CAC अगर हम ढूंढते हैं तो this is the amino acid histidine. So by that definition, valine will be synthesized, histidine will be synthesized. अच्छा, हमारा अब नया कोड आके यहाँ खत्म हो रहा है इधर. right? Because we are using this letter to join with this because this has been eliminated. So now our new code will be what? TGA. TGA. अच्छा, TGA को ढूंढते हैं हम. TGA is your stop codon. So since TGA is a stop codon, so your amino acid synthesis will stop, right? So this means that this mutation, which has been given as an example to you over here, is a deletion mutation in which one base pair at position number 6 is deleted. So much so that the reading frame is changed. कैसे? कि पहले दो amino acids के लिए तो it is not changed, but the amino acid ahead of that, जो कि पहले it was, representing some amino acid, now it has been changed from CTG. CTG कुछ था, क्या था CTG? देख लेते हैं नीचे क्या था. CTG. CTG was leucine. ठीक है, तो अब leucine नहीं बन रहा. CTG has been changed to TGA, and TGA is a stop codon, so your amino acid synthesis will stop. ठीक है, so let's look at this answer. इस वाले को part 1 का आंसर देख लेते हैं कि if the base pair at position number 6 is deleted, the polypeptide chain will be only 2 amino acid long, which is what? Valine and histidine. Why? Because after that we are ending up in a stop codon. Yes. Is this understandable? I'll continue the rest of the question afterwards, but please let me know, was this understandable or it went on top of your head?
So it was understandable.
Ji, Alia, what about you, beta?
Oh, yes, Miss. Explain.
Okay, and we'll be doing more difficult questions as far as mutations is concerned. Now, the topic of mutation is done, but I think in the next class, I'll be just wrapping up the entire chapter once again. I'll be doing some past paper questions. I would strongly advise you that you should go through the chapter thoroughly, check whether you have not understood any concept, because बीच में हमारा काफी gap आ रहा था. And inshallah, this Saturday, I will be taking an extra class. At the same time, please don't forget to join the class, right? So that we are up to the mark. Okay? So, see you on Wednesday, inshallah.
Miss,
Ji, Ahmad.
I have a question.
Yeah, sure.
ये जब TGA है तो ACT भी है तो फिर आगे स्टॉप क्यों हो गया? मतलब एक कोडॉन तो है, ACT बन रहा है.
एक सेकंड, TGA पे स्टॉप क्यों हुआ, ये पूछ रहे हो आप?
मतलब हाँ, ऊपर वाले में ACT भी तो है ना?
हाँ, very good question. Very ऊपर वाले में. We only read one strand of DNA. Can you see I've written this strand is the readable strand. ठीक है, तो एक ही strand read होगा, दोनों नहीं होंगे read. Are you getting what I'm trying to say? We can only अच्छा read. हाँ, when I say कि I can read a strand, reading का मतलब ये है कि I can only transcribe that strand into mRNA and I can only translate that strand into mRNA. For example, I'll just show you this. Let me just show you, because it's in the old textbook. This one. There is this chapter. Look, this diagram was actually really good in the old textbook. Now see, there are two strands, okay? One is running in the 5 to 3 prime direction and the other is running in the 3 to 5 prime direction. आप एक ही strand को पढ़ सकते हैं. ऊपर वाला strand ठीक है, उसका complementary strand तो है, but you can't read this strand. So this means only this strand is important, like one of the strand is important as far as the synthesis of polypeptides is concerned. So you would say that this is the readable strand. Okay? Or you can say that this is the sense strand because it makes sense. Are you getting what I'm trying to say?
Yes, miss.
Okay? Acha, have you done? हाँ, बोलो.
तो वो क्वेश्चन के अंदर हम जैसे उससे पहले वाला हमने I don't remember the letters, लेकिन उससे पहले वाला ऊपर वाला था. तो हम किस तरह डिसाइड करेंगे कि कौन सा वाला होगा, मतलब कौन सा सेंस बन रहा है?
हाँ कौन सा सेंस बन रहा है, दैट्स अ वेरी वेरी रेलेवेंट क्वेश्चन. इसको डिटेल से अगली दफा कराऊंगी मैं, राइट? बिकॉज आई हैव समथिंग, हाँ आई विल डू दैट इंशाल्लाह, बिकॉज दिस नीड्स अ लॉट ऑफ एक्सप्लेनेशन. योर क्वेश्चन इज वैलिड 100%, right? So we'll do that inshallah
Yes,
next
yes.
time.
Inshallah.
Okay, okay. Allah Hafiz.
Allah Hafiz.
Thank you,
Allah
Miss.
Hafiz. You're welcome. Allah Hafiz.